So hello friends, today we are going to discuss the penicillin. Okay, so first of all we will discuss the structure of penicillin. Okay, and its SAR activity means structure, reactivity and relationship. Okay, and its mode of action. Okay, so all of you know that it is a potent antibacterial drugs. Okay, and in this I have mentioned the structure of that. Okay, but before discussing the structure we want to discuss the some short story of penicillin, means some basic point, historical point of view. It is discovered in 1928 by Alexander Fleming, and it is isolated from Penicillium notatum. Okay, so it is isolated from Penicillium notatum. Okay, but in it, since it discovered in 1928, but in 1941 it is launched in the market. Okay, so basically, if you see in the industry, it is isolated from chrysogenum, Penicillium chrysogenum. Okay, so it is very important point for the discovery of penicillin. Okay, second is the we will describe the structure. So if you see the structure, I have mentioned the structure of penicillin, that it is the structure of penicillin. Two ring is present in this. Okay, one is one is lactam, and one is thiazolidine. Okay, so thiazolidine is 5-membered, while the lactam ring is 4-membered. Okay, so in this lactam amide bond is present. Okay, since it is in situ, means it is within the ring, so it is called lactam. Okay, so it is the definition. So first question arises from that, what is the lactam? So if you describe the lactam, then you can easily explain that if amide is present within the ring, okay, then it is called lactam. Okay, so one lactam is present, one amide bond is present inside the ring. Okay, while another amide bond is present outside the ring. Okay, and this amide bond, this carbonyl containing the R group. Okay, so it is R group, may be variant depending on the type of penicillin. Okay, so this one is thiazolidine, which is very important for the biological responses, and B is lactam ring. Both ring is very important, but most lactam, this beta-lactam is more important for antibacterial properties. Okay, so I have mentioned here that B is the beta-lactam ring, which is crucial for biological activity. Biological activity means antibacterial activity. Okay, and this amide ligand, since within the ring, it is called lactam. Okay, 2-methyl ring is also present, and it is each means each other is anti to each other. Okay, one is COH. Okay, so so if you see here, in this 2 ring, nitrogen is present, means in a bicyclic, means core of bicyclic ring. Okay, it is the bicyclic ring, it is the core in which nitrogen is present. Okay, so it is the structure of penicillin. If you see in this, I have also discussed the IUPAC nomenclature of penicillin. So if you see here, I have mentioned this chain of, means main framework of penicillin. And if you see the nomenclature of this, since it is USA nomenclature, so in this I have started from 1, means I have priority 1 is given to the nitrogen, 1 2 3 4 is given to sulfur, 5 6 and 7. Okay, so you can easily analyze that the IUPAC nomenclature will be the 4th thio, means on the 4 position sulfur is present, 4th thia, 1 aja. Okay, in one position aja is present. Okay, bicyclo means 2 ring is present, and it is 1, means bicyclic is how you can recognize this is bicyclic or not. Okay, so like that, if framework is present, okay, it is the ring, it is the ring, and this is the carbon, and maybe means you can replace one carbon by any other heteroatom. Okay, so the system will be the bicyclic. Okay, so it is called 4th thia, 1 aja. Why 4th thia, why nitrogen is not here? Because sulfur is priority will be given to the sulfur. Okay, so if you see 4th thia, 1 aja, bicyclo, number of carbon is 7, total number of carbon is 7 including nitrogen, so it is heptane. Okay, so 4th thia, 1 aja, bicycloheptane. But so it is the USA nomenclature, means in USA it is this nomenclature is followed, but in Indian system, Indian pharmacopoeia, it is called penicillin penicillinic acid. In this, methyl is added, extra COH is added. Okay, so these two fragments is added here in Indian pharmacopoeia for giving the IUPAC nomenclature to the penicillin. Okay, so it is common name is penicillinic acid. Okay, so it is very important point for IUPAC nomenclature. Okay, if you see the another point, a very important point, this lactam is very sensitive to H plus, means acidic condition. Okay, so if I actually this point I have explained here. Suppose that in acidic medium it captures the oxygen because it is electron deficient side, it is electron efficient side. Okay, so these hydrogen will be captured by oxygen, and this by capturing this tautomerizes and converted to this nitrogen. Okay, this nitrogen feels instability by which any enzyme, mainly bacterial enzyme, two enzyme is very important. One is penicillinase and other is beta-lactamase, which break down this nitrogen carbon bond. Okay, so it okay so okay, so for preventing this H plus from adding to this oxygen, means more electron reside, various types of penicillin designed and synthesized. Okay, so if you see here in this I have mentioned the one penicillin in which R is this one, benzyl. Okay, so in this hydrogen, two hydrogen, this is the alpha hydrogen because it is nearer to carbonyl group. Okay, and in this can be degraded by the any lactamase or penicillinase enzyme, but if these two hydrogen will be replaced by any steric, means any bulkier group, when this hydrogen will be replaced by this bulkier group, it feels the steric hindrance. Steric hindrance means it will prevent these beta-lactam moieties for degradation. Okay, so it forming the layer. If you suppose that any ring is you are going to add, then it prevents this one ring. Okay, so it covers this like that. Okay, so because if you add the substituent, it causes the steric hindrance. Okay, and it will cause the resistant nature. Okay, and resistant from where? Lactamase and penicillinase. Okay, so they protect the beta-lactam ring. Okay, so it is, so by which there are several penicillin is synthesized. Okay, for preventing this bacterial enzyme for degradation of beta-lactam ring. Okay, so there are various types of penicillin in which ortho, suppose that this is the benzene ring, this one, in this ortho position is blocked and meta is also blocked, but para is no effect of para position, means para position have no effect on the penicillin. Okay, for this property. Okay, so it is very important, and these are mainly there are various penicillin in which R is methyl group is added. Okay, so okay, so it is very important point. The third point is that I have discussed the structure, IUPAC nomenclature, this moiety, core moiety, and one is thing pharmacophore. So in penicillin, this moieties act like a pharmacophoric moiety. Okay, pharmacophoric moiety means it is responsible for binding to the receptor sites. Okay, when because it binds to the receptor site, it will show the pharmacological responses. Okay, because any moiety if going to attach any receptor is proteinaceous nature. Okay, when it combine to each other, they shows the any biological responses depending on the drug which are entered inside the body. Okay, so this pharmacophoric moiety is very important for biological responses and act here as a pharmacophore. Okay, so if anyone asks from you that what is the pharmacophoric moiety in the penicillin, then this ring is act as a pharmacophoric moiety. Okay, so it is very important. The another point is that if you see this one, so this is the structure. If you see the SAR activity, structure activity relationship, okay, then this moiety is present. Okay, this three group is very important. First one is this one. This hydrogen always remain in the cis position, cis configuration. Okay, if you add the trans hydrogen, then it will shows no effect. Okay, no means it will be the, it will not show the antibacterial properties. Okay, so the hydrogen will be cis configuration. Okay, and these methyl will be each other anti to each other. Okay, while COH, it is the acid by which penicillinic acid is given, nomenclature is given to penicillinic acid by this acid group. Okay, and it is responsible to binding to enzyme. Means if anyone asks which group is responsible for attaching to the biological enzyme, then COS, then acid group is responsible for binding to the enzyme. Okay, by which any pharmacological responses will be created here. Okay, so this will means this hydrogen will be cis configuration, this acid moiety is responsible for binding to enzyme. Okay, and this methyl group balancing the this moiety. Okay, so the ion and this two methyl will be anti to each other. Okay, the another point is that if you see the classification of penicillin, so penicillin is various types like G, V, K, F depending on the core, this R group. Okay, so it is depending on the difference in the R, it will named as GVKF. Okay, the second one, extended spectrum. There are various types of penicillin in which amino, carboxy and uride group is added. So these group will be arising in the spectrum, either they are proton NMR or proxy13. Okay, and Koji also determine these groups. Okay, so mainly these groups can be identified by the proton NMR. Okay, the third one is beta-lactamase resistant drug, beta-lactamase resistant penicillin. Okay, these are resistant to, these are the drugs which are resistant to the there will be no effect of any enzyme, either any bacterial enzyme on the beta-lactam ring. Okay, so they are resistant to any enzyme. Okay, the third one is beta-lactamase inhibitor. Okay, so these means they inhibit the beta-lactamase or penicillinase enzyme. Okay, by which beta-lactam ring will be not degraded. Okay, so it is a type of penicillin. The third one is, we will discuss here the mechanism of action, how penicillin act on the cell wall of bacterial cell. Okay, so before knowing this mechanism, you should be know the basic structure of bacterial cell. Okay, so these are the bacteria, these are the gram positive, this is the gram negative. Okay, so gram positive bacteria, if you see this is the glycan layer, this is glycan part, and this is the peptide bond, peptide ring. Okay, so these two forming the peptidoglycans. Okay, these peptidoglycan is very thick in the gram positive bacteria, while in gram negative it is thinner. Okay, and in gram negative outer cell wall is surrounded by a very thick layer. Okay, by which penicillin is not active against the gram negative, while it is much more active in gram positive. Okay, but in this exception is there, because in some gram negative bacteria there will be porins. Okay, porins is present in outer side of inside this ring. Okay, so that's why penicillin in some bacteria will be entered through these porins and will show the positive responses. Okay, but in this there will no extra ring is present outside the cell wall, so that's why penicillin is more active in gram positive bacteria. Okay, so these peptidoglycan provide the rigidity to the cell wall. Okay, because it is very thick and provide the rigidity to the cell wall. Okay, and so that's why its synthesis will be very important. But what happened here, I have also missing one point. This species is called periplasm. Okay, so if you see, the percentage of peptidoglycan is much more, because it is thick layer and as compared to this one. Okay, as compared to gram negative bacteria. Okay, so first of all, if you see here, sorry for that, if you see in cell peptidoglycan synthesis, the two amino acids NAG and NAMase have a very important role have very important role, and it is a carrier for amino acids from, means it carries the amino acid from cytoplasm to where, cell wall. Okay, so suppose that this is the cell wall. Okay, and in this NAG and name is present here. Okay, so cell wall is bilayered. Okay, so if you see, not in this I will discuss again in this. Okay, so you should be know that it is the, I have mentioned this, it is the cell wall layer. Okay, it is the cell membrane, it is the peri in between layer. Okay, you can say it is the periplasm. Okay, so in this NAG and name is present in the cell wall. Okay, and the full name of NAG, N-acetyl glucosamine, while this one name is N-acetyl muramic acid. Okay, so it is the carrier for amino acid. Okay, for carrying the and distribute the amino acid from one side to another. Okay, so since you should be know about that, in cytoplasm ribosome is present. In this protein is synthesized. If protein is synthesized, they form the amino acids. Okay, so, okay, not for that, sorry for that. It in cytoplasm, what happen here, ribosome is present. In this amino acid synthesize the protein. Okay, these actually there are 5 amino acids. Okay, NAM having tendency to attach from 5 amino acids. Okay, so if you see here, this one, okay, so these NAG and NAME. Okay, NAME is always attached from 5 amino acids. Okay, so and okay, so if you see here, in what happened here, in cytoplasm they go to through NAG and NAME go to, from where it carries amino acid, it carries from cytoplasm. Okay, so it is go to the cytoplasm and there amino acids present. Okay, and these amino acid synthesis the protein and where ribosome. Okay, so amino acid, 5 amino acid will attach from which mighty name. Okay, so it carries to tendency to capture 5 amino acids. Okay, and after that NAG is attached to that. So NAG and NAME is attached to each other, and after that they carries these amino acids to the cell wall. Okay, like that. This is the cell wall, this is the cell membrane. Okay, this is the periplasm. Okay, so after this they will, and in this if you see, these NAG and NAME come here and join to each other. Okay, this NAG and NAME and this NAG and NAME, in this NAG and NAME forming the CONH, NH2 present in the where NAG, while CO is present in the NAG. Okay, so sorry, NH2 present in the NAG while NAME is present in the COH. Okay, so if you see here, there will be formation of CONH. Okay, so this is very important for protein because this is the peptide bond. Okay, but what happened here, if penicillin target these 2 proteins, first of all, actually transpeptidase and D-L-carboxypeptide, 2 enzymes play very crucial role for synthesis of this one. First of all, transpeptidase replaces 1 amino acid from NAME. This is NAME. Okay, and this is 5 amino acid. 1 will be replaced by the transpeptidase. After that, D-L-carboxypeptidase forming the CONS bond. Okay, in this like way. Okay, so what happened here, penicillin targeted these 2 enzymes. Okay, so these 2 enzymes is there, these target the transpeptidase while another is D-L-carboxypeptidase. Okay, so these will be inhibited by the penicillin. Okay, so penicillin inhibited transpeptidase and D-L-carboxypeptidase, by which they will not cut the 1 amino acids, by which CONS bond will be not formed, by which peptidoglycan will be not synthesized. Okay, so this is the exact mechanism of penicillin, how they work on the cell wall. Okay, so they prevent the cell wall. Okay, since it is the essential component of cell wall, NAG and NAME is very important for forming the peptidoglycan. So that's why, okay, rigidity, which is responsible, this peptidoglycan is responsible for rigidity, means it provides the rigidity to the cell wall, by which peptidoglycan will, synthesis of peptidoglycan will be inhibited once time. Then what happened here, the cell wall will be burst. Okay, so that's why it is also called bactericidal. Okay, bactericidal are that agents uh, which inhibit the cell wall synthesis. Okay, so it is also called cell wall inhibiting agent. Okay, cell wall active agent or cell wall inhibiting agent. Okay, so this is the mechanism of action, how penicillin work on the cell wall. Okay, so it is the, I have discussed one point, historical point of view points. Okay, structure and its means SAR activity and mechanism of action. Okay, so it is very important for pharmacy students as well as medical students and medicinal chemistry students. Okay, for knowing these main points. Okay, so it is all about. And if you like this video, please subscribe my channel, and we will upload in the next any of drug like that. Okay, and we will describe the structure, same property we will describe here. Okay, so thank you for watching this video. Okay.